Thanks so much, Willie. And Bill, it is a pleasure to talk to you today. And actually, I think one of our most important discussions, because the theme of this forum is how to make medical progress faster. But unless we know kind of what the path, where the path is leading when we say progress, it's kind of hard to get a full picture of why faster is important. And I know you've talked about a world without disease. So it's kind of hard to hear that and not feel in a way that it's a little bit hyperbolic. Is that the right word? Hyperbolic. So I'm wondering if you could just explain a little more about your vision.
No, I'm happy to, Ellie, and it does sound that way when you hear it. But I like to start by asking everybody who would like to get a disease. Please raise your hand. I've never seen anyone raise their hand. And in my experience of 30 years practicing medical oncology, never had a patient once come in and say, Dr. Hait, I'm so happy that I've been diagnosed with cancer so you can treat me. Yeah. So if you really want to make transformational progress, you would look to prevent diseases or intercept disease-causing processes before anybody got ill. And the issue that we face is that the very practical issue that patients are coming in and they're in front of us and they have immediate needs. So much of our time and money and attention correctly has to pay attention to those immediate needs. But at the same time, I think we could do more than we're currently doing to study how to prevent and how to intercept diseases so we don't have the acute problem of so many people suffering with so many diseases.
And when you say intercept, do you mean diagnose early? What would interception look like?
A good example of interception, I'll give two. I'll give one in the cardiovascular space. So this is well known to everybody. If you have hypercholesterolemia and your LDL is too high and your HDLs are too low, you don't wait until you have a heart attack or a stroke to treat the heart attack or stroke. You intercept the process by going on a statin or a PCSK9 inhibitor. And that's a good example of interception in the cardiovascular space and one of the most successful. The other might be in the oncology space, where every cancer has a precancer, a premalignancy. Breast cancer has ductal carcinoma in situ. Multiple myeloma has smoldering myeloma and MGUS. So rather than wait, we don't take someone who has ductal carcinoma in situ and wait till they have invasive breast cancer to do something about it. We intercept the process. Right. Wouldn't it even, and same thing now with smoldering myeloma, we don't wait until people's bones are crumbling from multiple myeloma. We treat, and that's relatively new. And as we get closer and closer to understanding the origins of these diseases, our ability to prevent or intercept them, or ultimately by early diagnosis to cure them, will become much more of a reality.
And so what's missing now? Why aren't we there now? Is it some combination of basic and translational research, or?
I think there's many different things that could and should be done, but fundamentally we need more knowledge. We need to understand, why does someone develop ductal carcinoma in situ? Why does someone develop MGUS or smoldering myeloma? Why does someone develop Parkinson's disease? What's causing these things? So fundamental knowledge. And with fundamental knowledge comes the ability to understand targets. So a great example would be SARS-CoV-2 virus and COVID-19 disease. We very quickly knew what caused COVID-19 disease. It was the SARS-CoV-2 virus. And then we very quickly, based on the sequence, knew how that disease was being caused through the spike protein interaction with the ACE2 receptor. And then it was became feasible to make monoclonal antibodies and to make vaccines because we had the knowledge. And look how quickly that was done. Almost miraculous.
Yeah, it seemed, incredible how quickly it happened. But the process, in a sense, the process was still there. You had to have that causal piece at the bottom of, and then build from there.
You had to have knowledge. And that's critical.
Bill, let me switch gears to another topic we've talked about, and that is an innovation model that J&J is trying. That it seems to me it's almost like all in. That you're looking for ideas where they are good, not based on somebody's maybe degrees or their or which university they're doing research from. Could you talk a little bit about your innovation model and your goals for it, J&J's goals?
Sort of the tagline for Johnson & Johnson Innovation is sourcing innovation wherever it originates. And we recognized some years back that no matter how large a pharmaceutical company or healthcare company is, or how much R&D budget you expend, you can't possibly capture all of the innovation, the spectacular innovation that occurs around the world. You can't just possibly do it within your own laboratories. So we very quickly set up a system to make certain that we were well positioned in the innovation ecosystems. So San Francisco, Cambridge, Massachusetts, London, England, Shanghai, and many other places around the world, we put innovation centers. These are basically partnering offices where we have experts from our internal businesses, R&D areas, living in those ecosystems and getting to know the innovators. And they're supported by finance people, by legal. So we could move rapidly to create an early deal rather than having to go back to corporate headquarters and get in line with some of the later, more expensive deals that take up a lot of time and energy. We also created incubators around the world. We call them J-Labs. And we've incubated over 700 companies so far. And we have hundreds of collaborations with those companies to get entrepreneurs started, to help them by creating the infrastructure for them so they don't have to buy expensive mass spectrophotometers or cell sorters or things that are very expensive. And then finally, we have a venture fund called JJDC, and there we use our corporate venture dollars to help invest in, support these new innovative ideas, these new corporate companies that are starting to support entrepreneurship with the ultimate goal of sourcing that innovation for our pipeline. Although I must say that others have taken advantage of the incubating companies as well, which is fine if there wasn't a high priority for us.
But what's so great is the infrastructure you're building, right? So those companies are not going to necessarily be one drug company or one solution company, one diagnostic company, right? So you're actually letting 1,000 flowers
It's exciting
bloom.
to see. It's so much fun. To visit the J-Labs or the innovation centers and meet with these young people who are so excited, so passionate about their ideas. And they'll often turn to me and say, after they tell me about it, so Bill, do you think it's a good idea? And I would quote an old Yale professor, Bill Prusoff, and I would say, if it works. They're loaded with energy.
Yeah, I'll get back to you.
Loaded with ideas. It's spectacular to see. And I think it gives all of us hope for a healthier future and getting us closer to a world without disease.
And Bill, is it also, we haven't talked about this before, but is it also part of a DEI, built into your DEI strategy? It seems like diversity would come naturally when you're really allowing folks with good ideas instead of choosing folks and then hoping they have good ideas, that you have to bring diversity in. Is that happening?
Actually, one of the, for our strategic plan for Johnson & Johnson Innovation, one of the objectives is DEI. And we have a very interesting and very important way, I think, to help with DEI, because many biotechs, academic investigators, they would like to create a partnership with us. And that's great. We're thrilled by that. But one of the things we always bring up now, I think certainly more intensely over the last couple of years, is we want to know their commitment to diversity, equity, and inclusion. Is it consistent with ours? Are there members of their leadership team from diverse backgrounds? Are there products that they're working on? Is there a thought? Is there thinking about how equitable the distribution, the availability of those products would be? And we just bring it up as part of the conversation. And I think by hearing
That's
it
terrific.
from, a very large healthcare company like Johnson & Johnson, it has some impact.
I love that, because again, it goes back to infrastructure, right? You're building that value into the R&D infrastructure, and you are a very large and influential company. And so that can have really dramatic impact. Okay, now I want to ask a hard question. Tell me about something that went awry. It can be a good idea you had, but it wasn't at the end, if it works. And maybe it didn't, or just something where you learned a lesson in what you're doing with innovation, or what you're doing with this model of a world without disease.
Yeah, I think that there's too many. I'd have to go through a whole list of thinking, going even back to my days where I ran a relatively good-sized laboratory back at Yale, then at Rutgers. We would really want the students and postdocs to come in and tell me, well, what is your big vision? What would you like to accomplish? My PhD advisor said to me, don't read anything. Just think about what you would like to do.
Just say it.
And, and we would think about these things. So I started, when I, this goes back a little bit, Ellie, from my PhD, where I was just supposed to think of something, and I wanted to cure cancer. So that's a long-term passion. And I was absolutely certain in those days, of course, 40 years ago, I was absolutely certain about everything. Today, I'm less certain about anything. I said, I knew that the key was signal transduction. And in those days, the only signal transduction pathway known was cyclic nucleotide metabolism. So, of course, I said,
so what signal, I'm so sorry, but what's signal transduction?
Extracellular, something outside the cell tells something inside the cell what to do. Yeah.
Oh,
so
what
it had
to do.
be cyclic nucleotides. And so I decided I would cure leukemia by studying cyclic nucleotide metabolism. So it turned out that there were reason to suspect, and we actually stumbled on a few things, like the enzymes that break down cyclic nucleotides were completely altered in cancer. And then we found that you could selectively inhibit one molecular form versus another. And then we tried it in leukemia models, and they didn't work at all. Didn't work at all. However, others over the years have used the same approach. And the first drug, for better or for worse, was actually the first drugs for impotence, worked by inhibiting phosphodiesterase PDE5. So we never thought that Viagra would be the first one. We were trying to cure leukemia, but I guess it has a very important role in the world, and so some contribution.
But it's so cool because it shows that, failure isn't failure in science,
Yeah, no,
right? you uncovered new knowledge, in this case, really valuable knowledge for a lot of people.
I must say it was worth many, many, many hours in a cold room trying to get the columns to behave to get there. But it was a wonderful experience.
Very cool. I am very sad because I think we're out of time. I feel like I could talk to you forever, but I always feel that way. So thank you for joining the forum, and we'll be in touch.
Thank you for having me. It was my pleasure.
Thanks.